Hi, I'm Jay Phelan. Let's talk about some biology. We're going to continue talking about linkage today, and because it's such an abstract concept, it's helpful to use some examples based on real-world data and seeing what we use these data for and how our understanding of linkage helps us to better understand inheritance. So we always start from Mendel. Remember, he had this concept that genes were free-floating entities in the cell, hereditary factors, he called them. There were two copies of each, one you got from your mother, one from your father, and his law of segregation says only one of those copies was passed down into the gamete. Based on his conception of inheritance, that they're free-floating in the cell, he expected independent assortment. This is his second law that says the pattern by which you inherit one gene is not going to influence the pattern by which you inherit another. Everything behaves independently. Therefore, if you were to look at two genes simultaneously, when you did a cross of individuals who were heterozygous for both, the ratio of phenotypes that you would see would be 9 to 3 to 3 to 1. 9/16 having the dominant trait for both genes, 3/16 having the dominant for one, recessive for the other, 3/16 having the reverse, and 1/16 having the recessive trait for each. But what we see is that this doesn't always happen. And Reginald Punnett did a cross where he was also working with peas, and he was looking at purple and long versus red and short, and he did a double heterozygote cross where you're going to get 4 different categories of offspring, purple and long, these are the dominant, that's why I have them in all caps, and red or round, those are the recessive traits. When you do this cross, what do you expect the ratios to be? Well, Mendel would expect them to be 9 to 3 to 3 to 1. But when Punnett did the cross, he found this. He had 381 offspring that he looked at, so you have to figure out what 9/16 is of that. So it's about 215, 71, 71, 24. But his observation showed that there were way more of the purple long and the red and round than he expected. There were far fewer of the purple round, red long, than he expected. So we got more double dominants and double recessives. So that first take-home message we had was that often we don't see independent assortment. So Mendel's law, as a general rule, pretty good, but there are many exceptions. Then we looked at T.H. Morgan, Nobel Prize winner for studying how chromosomes were related to inheritance. He looked at fruit flies, looking at the trait vestigial wings, so it's kind of a shriveled up wing as opposed to the normal one, and he looked at purple versus red eyes. But it was the same idea. Look at two traits, see what happens when you've got this double heterozygote. And he did a couple of different setups, parental generations, in order to get those double heterozygotes. So he had red and normal wings, red eyes, crossed with purple eyes and vestigial. Purple and vestigial are recessive, red and normal are dominant. And so when you do that cross, you end up with all red, normal winged flies. But he did another one where he had purple eyes and normal wings, and he crossed them with red eyes and vestigial wings, and same thing. You still get a double heterozygote, shows all red, normal, and the genotypes would be described as VG, VG+, PR, PR+. So he did those crosses, but then, this is where he had the clever idea, was this. If he test crossed those F1s, instead of doing F1 crossed with F1 to get the 9 to 3 to 3 to 1, he said if I test cross those F1 offspring, the results are going to tell me what gametes that double heterozygote is producing. And this is really clever, and we're going to look at it. So we've got all red normal, but they're an F1. So we know what they're going to be heterozygous, PR+, PR, VG+, VG. When you do a test cross, remember that's crossing it with an individual that is homozygous recessive. Here the plus stands for the wild type. It's dominant in both of those cases. So you do the test cross, and the test crosser is always going to contribute the same gamete, the alleles, same alleles in the gamete. It's going to be the recessive for PR, the recessive for VG. On the other hand, our F1, the doubly heterozygous one, is going to contribute maybe a PRVG, maybe a PRVG+, PR plus VG, PR plus VG plus. So those are the 4 options. And if you look at the phenotypes, when you get the results of this cross, you would expect equal numbers of all of them, because we think that that individual, the doubly heterozygous one, is going to produce equal amounts of all of the gametes. But what we found, instead of our expectation of equal numbers of each, Morgan observed tons of two types and very few of the other. So something was going on. And he concluded that the genes are on the same chromosome and they're close together. And because they're close together, they're inherited not in an independent assortment fashion. If you visualize it with a line to represent a chromosome, now you've got the maternal copy and the paternal copy of the chromosome. So we put the PR and the VG on one of them, and the PR plus and the VG plus on the other. But now we've got a line holding them together. So then when you make a gamete during meiosis at anaphase, when you pull apart, separate the homologs, the PR plus and VG are stuck together. So the gamete gets both of those. The PR VG goes elsewhere, and those are stuck together. So we'd expect only those types of gametes rather than all four. So if you have linkage, the double heterozygote should only produce two types of gametes, unless there's some recombination between the homologs. If there is, then every time you get a crossover there, as I've drawn in at the bottom here, a crossover between the genes, then the new linear strand is going to have the allele from the maternal homolog and the other gene's allele from the paternal homolog, and vice versa. You create equal numbers of each. So now we'd also get a PR plus and a VG, a PR and a VG plus. We just wouldn't get as many. So let's look at an example. Say you've got a mouse that's got a genotype big A, big B, little b. We're looking at two genes, heterozygous for both. And I'll introduce a new phrase here, coupling linkage phase. What that means is that the big A and the big B are stuck together on the same linear strand of DNA, the same chromosome. Maybe it's the maternal homolog, maybe it's the paternal. And the little a and the little b are together. This would be in contrast to what we call repulsion linkage phase. And so people will just refer to that to tell you how those initial double heterozygotes were created. So back to our problem. We say a mouse has the big A, big B, little b. It's coupling linkage phase. So we can draw it out. The big A and the big B are on one of the homologs. The little a and the little b are on the other one. What gametes are going to be produced? Well, the big A and the big B are going to be together. The little a and the little b are going to be together. So we'd write it out as big A, big B, a, little b, just to note those go together. Now we're going to do what Morgan did. We're going to do a test cross of this doubly heterozygous genotype times an individual that's little a, little b, little b. The test crosser genotype, the little a and little b are going to be stuck together. Another little a, another little b are going to be stuck together. The gametes there are all going to be AB, little a, little b. So we know that. So let's look at what happens here. Mendel would have expected from a double heterozygote 4 different types of gametes in equal proportions. One type of gamete, all the same for the test crosser. But in the absence of recombination, that big A and big B, they're stuck together. They go together. The little a and the little b, they're stuck together. They go. And you can't get the other one, the one that has one of each. So we would expect to get half of each. So the result of the test cross would be half of them would be doubly heterozygous, showing the dominant phenotype for both genes. The other half would show the recessive phenotype for both genes. They'd be little a, little b, little b. But we'll add in something here now. We say recombination does occur. So we're making it more realistic. And we say there are 20 map units between the genes. A distance, a physical distance. Chromosomes are a physical thing. So now we're saying, here's how far apart they are. Why would we care how far apart they are? Well, crossing over is equally likely to occur at any spot on the chromosome. Therefore, the further apart they are, the more opportunities for a crossover to occur. So what gametes would get produced here? Well, we'd see big A, big B, big A, little b, big B, little b, all 4. But because we have them linked, we're not going to see 25%, 25%, 25%, 25%. The parental gametes, they're called, those are the ones that are the pair of alleles that are on the same stretch of a homologous chromosome here. So the parental ones would be big A, big B, and little a, little b. The recombinant gametes, you have to draw in, okay, we had a crossover occur somewhere between A and B. The recombinant gametes would be big A, little b, big B. And here's the interesting thing. 20 map units tells you that 20% of the gametes that you're going to see from this doubly heterozygous individual are going to be recombinant gametes. That's what the distance is. The distance is defined as how often does crossover occur between them, because I just said it's equally likely at any spot. So if we say 20 map units, that means 20% of the gametes have to be recombinant gametes. So how many do we have of each? Well, it's easiest to start with the recombinant gametes. 20%, it's going to be evenly divided always between the two because each crossover event creates equal numbers of the two different types. And because it has to total 100%, we've got 10, 20%, so there's 80% left. Therefore, it's got to be 40% of each of the other two types of gametes. Contrast this with a situation where A and B are on different chromosomes, or A and B are so far apart that they don't behave as if they're linked. In that case, you get 25%, 25%, and so on, if they're on different chromosomes. We call that unlinked. So our take-home message here is that the map distance, a physical distance, is defined as the percentage of gametes produced that result from recombination. Now think about, so I've already said that there's a relationship between the map distance and the proportion of recombinant gametes. That's how it's defined. But think about why that is. Be able to articulate that, because that's an important thing, that we're going back and forth between a frequency of something that's produced and saying that that is a physical distance. So that's kind of a weird thing to be equating. Let's look at another example. Let's say we've got two loci and they're 15 map units apart. We cross two double heterozygotes in coupling linkage phase. So coupling phase means that the dominant alleles are going to be on the same homologue, the recessive alleles are going to be on the same homologue. But I say here we've crossed two double heterozygotes. So we're not doing the test cross this time. We want to know what proportion of genotypes and what proportion of phenotypes do we get. So if I say crossing two double heterozygotes, you should immediately be thinking, all right, there's going to be a 4x4 Punnett square with all the gametes that they can make down here and down here. So let's look. So here's what the cross is going to look like. I've drawn them in coupling linkage phase. I've just arbitrarily named the genes R and B. So we got a big R and a big B together and a little r and a little b. For both of these, you can see if you just wrote out the genotype without putting the chromosome in there, it would just be big R, big B, little b. They're doubly heterozygous. We do the cross, we think about first, what gametes are produced by each of these? Well, all four, but not in the 25%, 25%, 25%, 25%. Instead, we have the parental gametes, that's the big R and big B, and the little r and the little b. We also have the recombinant gametes, the little r, big B, big R, little b. And here's the percentage that we're going to produce of them. 7.5% of each of the recombinant gametes, because I've told you what the distance is. It's 15 map units. Divided in half, each of those 7.5%, that's what we get for the recombinant gametes. Whatever's left over is going to be divided between the parental gametes. So 85%, we divide that in half, 42.5% for each of those. Now, we know the gametes that each of these double heterozygotes is producing, and we know how frequently they're being produced. So the question that we had was, what proportion of genotypes and phenotypes do we get resulting from this cross? So we got to do our big modified Punnett square here. Female gametes go across the top. All four are produced, but I left them in the purple and the light blue to remind you that some of them are parental gametes, some of them are recombinant gametes. Same thing for the male. And you'd go through, and for each of the 16 squares, you'd say, okay, if that male gamete fertilized that female gamete, the genotype of the offspring would be big R, big B, big B. You could fill out all 16 of them, any of them. Let's say the third row, third across, you'd get a little r, big B from the male, little r, big B from the female, so the genotype is going to be little r, big B, big B. But in the past, whenever we did a 4x4 Punnett square, each of those 16 cells was equally likely. But now we've just said that the little r, big B, the big R, little b, those are not as common as the parental gametes. We can attach a probability or a proportion for each of these. So 0.425, 42.5%, are big R, big B. 0.075, that's 7.5%, are big R, little b, and so on. We do the same thing on the top for the female. Now every one of those 16 squares, it can occur, but now we can say, well, how likely is it to occur? And you say, well, what's the probability of getting that male gamete, the big R, big B? It's 0.425. What's the probability of getting the big R, big B gamete in the female? 0.425. So you multiply those together, and 0.181, 18% of the offspring are going to be doubly homozygous dominant. So we do that, then we go down to the next one. We say, all right, well, what about the next one? It would be that big R, little b male gamete occurs 0.075. And if the big R, big B for the female, 0.425. So we multiply those together, you get 0.031875, 3% of the time. You can do this for all 16. They're still going to add up to 1 because that's 100%, but now they're not equal. Certain genotypes are more likely to show up than others. And remember, the question that we asked was, what are the proportion of phenotypes? So you got to look which ones are going to show the dominant for R and the dominant for B, or the dominant for R and the recessive for B, and so on. So one aspect of that question is what I've got below here. What proportion of the offspring are big R, anything else, big B, anything else? In other words, typically we'd expect 9/16 of them to be that. So out of the 16 squares, 9 of them indicate a situation in which the individual is going to show a big R, whatever that dominant trait is, and the big B dominant trait. But in order to do that, you'd have to add up the frequency of each of the 9 squares that have a dominant R and a dominant B, independent of what the second allele is. It turns out to be 68% of them. Next question says, how does this differ from the unlinked cross? Well, in the unlinked cross, big R, something else, big B, something else is 9 out of 16. What's 9 out of 16? 56%. So we say, all right, we got more of the big R and big B showing up together. Well, that's because they were in coupling linkage phase. So the difference between this cross of linked genes and the unlinked gene is that certain genotypes in the offspring are going to show up more commonly. Certain traits are going to go together. And you see this in the real world. Hair color and amount of freckles. You see red hair and freckles goes together a lot more commonly than it doesn't go together. So that explains what is happening here. And it's helpful if you go through and you fill in the probability of each of these 16 outcomes occurring and adding them up and making sure that the numbers work out for you. So there you have it, an example of linkage and recombination.